Welcome back. This is the 5th video in this series on biological molecules. In this video we're going to deal with proteins. And proteins, as we mentioned with carbohydrates, we mentioned with lipids, proteins contain carbon, hydrogen, and oxygen, but they also contain nitrogen. Unlike the carbohydrates and unlike the lipids, they've got nitrogen, but also they're not used for energy. We do not normally, unless you're in starvation situations, our body doesn't metabolize protein for energy. They're made out of unit molecules called amino acids. And these do form true polymers. So the unit molecules are amino acids, the polymers are actually called proteins. So that old picture we had, and I can just put it here again, always come back to this picture. I've got 4. That's not very many. I'll put a couple more. Still not very many, but this would be a protein. And these are amino acids. And of course it can go both ways. And if it's dehydration synthesis, we get the water molecule coming out. So that's the pattern we're onto here. How short can they be if they're a protein? Well, you know, if you take 2 amino acids, you can put another amino acid on it. The shortest protein that I know is oxytocin and ADH. Those are 2. ADH is one that is produced in the brain actually and is released by your posterior pituitary gland. ADH is used in the urinary system. It has 9 amino acids. That's the shortest one. Oxytocin, part of the reproductive system in females, also has 9 amino acids. So these things, those are maybe the shortest ones. Insulin, 51 amino acids. That's sort of maybe a mid-length one. Some of them can be hundreds of amino acids long. Some of these things, if they're enzymes, this is a hormone, some enzymes can be hundreds long, so there can be hundreds of amino acids. But before we get into all that stuff, we really have to look at what the heck amino acids look like and how we can combine amino acids into these structures here. Now amino acids, I'll just leave the word right there. Amino acids all have a common structure. They all have N, C, C. That's the common backbone, if you like, for an amino acid. This end, like that. This end, like that. This is an amino end, or sometimes called the amino terminus. Well, terminus just means end. And this is the carboxyl end, or carboxyl terminus, the carboxylic acid group. Bonding requirements, think back now. Hydrogen has 1, nitrogen has 3, carbon has 4, 1, 2, 3, 4. Oxygen has 2. We need 2 more things on here. We have a hydrogen and we have an R group. And you say, what's an R group? Well, you think about these things, periodic tables. There's no R on the periodic table. There's no element that has this symbol R. The R really just stands for rest of the molecule. So what could R be? Well, if R is just hydrogen, so if we take this out and put a hydrogen in here, you recognize that's the smallest amino acid. It's got the NCC background, it's got all of these parts on it. The R group is just a hydrogen, that's glycine. That's the name of that amino acid. If we take this out and we put maybe the next smallest thing in here, we say, okay, the carbon is going to be bonded to well, we can put OH in here. That would be the next smallest thing, I guess. So if R was equal to OH, that's the amino acid called serine. Or if we went to a carbon and we put 3 hydrogens on it, so we have something like that, that's alanine. Or if we went bigger, you're getting the idea, I think, that there's going to be lots of different things we put in here. We go to another, I'll draw it differently. Go this way, carbon that way, we put a hydrogen in there and put CH3 out here and CH3 out here. That's valine. So that combination there, I won't try and draw it there, is valine. How many are there? There's 20. There's 20 different R groups. Which means, quite simply, there are 20 different amino acids. How complicated can they get? Well, fairly complicated. These things can actually have ring structures on them. So you wind up with, like, tryptophan, for example, has got cyclic components out here. So they get quite big. But anytime you see this part here, it's an amino acid. That part, with one exception, and I'll mention that exception before we finish this topic, with one exception, that part never changes. Never changes. Okay, so what I'd like to do now that we have a rough idea what amino acids look like, is I'd like to do this dehydration synthesis. How do we get amino acids to combine together? So I'll leave that there as a reference. I can take this out. I'm going to take glycine and alanine, I think. So we take glycine, which is, notice how I start, NCC. Glycine is the smallest one. And we're going to take alanine. NCC, and I think now as soon as I put these things side by side, you're going to see exactly what's going to happen here. Dehydration synthesis, we're going to get a molecule of water being given off. You can see where that's going to come from. It's going to be right here, not there, but here. Does this form an ester bond? No, because ester bonds, if you remember, went carbon-oxygen back to carbon. This is not doing that. This is going from carbon straight to nitrogen. This thing is called a peptide bond. A peptide bond is a strong covalent bond. Now if I redraw the thing properly, it's going to look like this. Now watch what I'm going to do. I'm going to do this on purpose here. I'm going to take this molecule, I'm going to put the N, I'll make it a little bit smaller because I don't want to run out of space. OH. I won't put all the hydrogen on. This bond angle is pointing down here, isn't it? So if I take this molecule and do it properly, I'm going to have the hydrogen from that nitrogen in here like this. There's the nitrogen, hydrogen, and this molecule is more like this shape. The CH3, because that's alanine. So in combination, when I get the peptide bond forming, the peptide bond forms between the carbon and the nitrogen. The point that I'm trying to make is that it's not linear. This is a bent molecule. And if we keep going with this, if we put another one on here, the same thing, it's going to bend it again. It's going to bend it again if you go on to the other one. So what happens with this molecule is it actually turns. And it does this, it actually forms a spiral, which is another topic that we're going to get to just in a quick minute here. But I'm going to tell you this. This thing here is called a dipeptide. two amino acids held together by a peptide bond, dipeptide. How many different dipeptides are there? Lots. Well, if you take this first thing here, we've got 20 different amino acids that could be there. I just used glycine as an example, and you could have 20 different ones there. There's actually 400 possible dipeptides. When we talk about variations, what if you had a tripeptide? 20 times 20 times 20? That's 800 possible different tripeptides. Because just because you use glycine doesn't mean you can't use it again. You gotta have glycine. So you get a protein, and this is what I was leading up to. Talking about ADH and oxytocin, 512 million possibilities. Right? If you do the math, 20 times 20, 9 times, 512 million possible combinations. ADH is one particular combination. And one of the things that's going to show up in future video is that if you screw that up, not you, but if it gets screwed up, if you get the wrong amino acid in the wrong place at the wrong time, you get a protein that doesn't function. It won't work. And if it's a hormone that won't work, or insulin won't work, or an enzyme, as I had down here when I was talking about enzyme, doesn't work, there's some metabolic disorder. And that's, like I say, that's another whole topic. Now, what I was alluding to, and I started getting into it, this thing here, once you put this stuff together, you see that not only can you have a sequence of amino acids, and like I say, the sequence can be, well, there's millions of possible sequences, but you also have a consideration where that sequence isn't a straight line, it actually turns. Those considerations are called levels of protein structure, and that's what I'd like to go to now. I'm going to erase this because I don't need it. Actually, I don't need any of this. Levels of protein structure. The first level, as it's called, is primary. So we talk about the primary structure of proteins, that is the sequence of amino acids. That's what that is. So which amino acids bonded to which amino acid and so on. And like I say, there's millions of possibilities there depending on how large of a protein you're talking about. Secondary structure is what happens to that sequence of amino acids. Now, if it was a straight line, that'd be one thing, but what I'm saying is not a straight line because of the bond angles that are there. You wind up with one amino acid, the next amino acid here, next amino acid, next amino acid. And what it does is it actually spirals. It actually takes 3.6 amino acids to make one complete loop in this thing, sort of like a slinky, maybe, or a spring. 3.6 amino acids per loop. Secondary, that's called an alpha helix. Alpha helix. A beta helix would be turning the other way, but this one turns this way. So in secondary structures, a beta helix. Now this is why I want to mention that other particular amino acid. Here we go with NCC, remember? They all have that. This part is always the same. I should have drawn that at a slightly different angle, but I'll fake it a little bit here. This and the amino group of this particular amino acid is caught up in a ring structure. There's a 5-cornered ring here, which means that this thing can't form a particular bond like it's supposed to. It hasn't got that possibility to form the peptide bond. So this amino acid, which is called proline, anytime it gets incorporated into an amino acid or into a protein, because it can't bond like the other ones do, it causes a kink. So if you get this thing like this, it's forming this nice spiral, and all of a sudden there's protein in it that goes out here. It might form a spiral over here again after the effect of proline isn't there again. Then you might get another proline that forms another one like that. So you get this sort of next level of protein structure. One of the things that can happen in here, just before we get into the next level completely, is these things can line up with each other. And there's another variation of secondary structure, and it's called a beta pleated sheet. So instead of the amino acids forming a spring-like structure, they actually lay side by side, and you get cross-bridging and cross-bonding in there. So there's two possible variations, but this is the fundamental secondary structure. I'm going to erase this over here in a minute here, and then we're going to talk about what kinds of things hold them together in those configurations. But let's get to this one, tertiary. This is where you get the 3D shape, this globular type thing. So if I were to take that effect of proline and continue it, so we get this, here's your alpha helix, all of a sudden it does this, and then it does that, and then it does this, and it does that. And the overall shape of this protein is, I can only draw 2 dimensions on a flat board, but you get this mass, and it's got a thickness to it as well. So this is the glob shape, which is a 3-dimensional configuration. And if that weren't enough, some proteins have another level of structure. And I say some proteins, let's just back up a tiny second. All proteins have to have this level of structure. They all have to have that. All proteins have to have this level of structure, because you get the sequence of amino acids, it's either going to form an alpha helix, which is normal, it usually does that, or you're going to get a beta pleated sheet. Some proteins, many proteins have this level of structure, but they don't have to. You could have a single protein which is just a linear sequence, something like the protein tubulin, which is the name of the protein in cilia and flagella, because it's just a long strand. It doesn't form that kind of a thing. So all proteins have these. Some proteins have this. Things like enzymes have this. Enzymes are tertiary because it's the shape of the enzyme that is important in how it functions. Some, fewer, have another level of structure, and it's called quaternary. And this is the association between tertiary structures. So you have a set of tertiary structures which function together. I just want to give you some abbreviations, because to write out tertiary structures and quaternary structures all the time is a lot of writing. Primary is often abbreviated just first degree of structure, secondary, secondary degree, third degree of structure, fourth degree of structure. So you can use those abbreviations, they're very well known. Example of quaternary structure, I'll give you 2 examples actually. Hemoglobin. Hemoglobin is a protein. Hemoglobin is the protein that's on the surface of red blood cells. It transports oxygen. The only thing is hemoglobin as a protein has 4 tertiary structures, but it has 2 different kinds of tertiary structures. So it's like it's got 2 alpha ones and 2 beta ones, so it's 2 of each. 2 of each. 2 of each of 2. So a total of 4. Another one is LAD. Now you probably haven't heard of LAD, but I can tell you what it is, and you will have heard of that. Lactic acid dehydrogenase. Lactic acid is that acid that builds up in muscle tissue that causes sore and stiff muscles if you overexerted yourself for a period of time. Lactic acid dehydrogenase it's an enzyme. It has 4 different components to it, 4 different tertiary structures. I said I was going to erase this, and we'll talk about the kinds of bonds that hold these things together. Take this away. I'm just going to erase, I'll erase that part. So we've still got the examples, we're okay with that. Primers, this is held together by peptide bonds. Very strong, no problem. These ones, these are held together by hydrogen bonds. Now I didn't get into that, but if you think about it, if you take that alpha helix, like that, there's OHs and nitrogens in here. What you get is dipoles, and the dipoles are holding this thing together. So you get hydrogen bonding forming between areas of opposite dipole along that spiral thing. So there's quite a few of those hydrogen bonds can form, because one can form with every single amino acid. So you get 3.6 amino acids per loop, you get 3.6 hydrogen bonds per loop. So that is a fairly rigid structure. This one, tertiary here, is held together in addition to that hydrogen bonding by hydrogen bonds again. But you can also have some ionic bonds and covalent bonds. Now let me explain how that happens. By the same reasoning, if you get different dipoles held close together, you can have hydrogen bonds forming in different parts of the molecule here. So you do get hydrogen bonding within that three-dimensional structure. But we also have other molecules or other atoms which are in proteins, in addition to carbon, hydrogen, oxygen. You can have, for example, sulfur in some amino acids, and you wind up with a thing called a disulfide bridge. So you can get another kind of, so maybe here we have two sulfur atoms are actually side by side, sulfur, sulfur, and that forms a disulfide bridge, which is a very strong covalent bond. So we do have some covalent bonds and some ionic bonds in that level of structure, as in addition to hydrogen bonds. And these ones are just held together by weak bonds, hydrogen bonds, attractive forces. They're held in association with a central group. Like, for example, hemoglobin, we have a heme group in the middle, which is where the iron is contained, and you wind up with 4 proteins, 4 tertiary structures associating with that heme group. So it's not very hard to destroy that type of molecule. It's also not hard to destroy this one. Hydrogen bonds themselves are weak. There's enough hydrogen bonding here that doesn't get destroyed too easily. But if we're talking about enzymes, we'll go back to this picture. If you heat up an enzyme, the first thing that's going to happen to it is these hydrogen bonds will break, because there's not very many of them and they're weak bonds. If those hydrogen bonds break, then the molecule can kind of unravel a little bit, and if it unravels, it doesn't work, because proteins are the shape they are so they can function properly. And we're talking about enzymes. If that happens to be an enzyme has to match with a substrate, and if it doesn't match in terms of three-dimensional configuration, three-dimensional shape, it simply will not work. If that happens, if we take away hydrogen bonding in here, these ones can be denatured. This topic is a big topic that shows up in another video. So we get into how we actually change the shape of enzymes, how we denature them, and the factors that contribute to denaturing. But it's worth noting here that it's the loss of hydrogen bonds, the weak hydrogen bonds in tertiary structure, that leads to them being denatured. I'll give you one example of how what that is, and you'll recognize this example. There's a protein called casein. That's a protein that's in milk. And as long as everything's fine, You don't notice it, you don't see it. But go ahead and denature it. Put a little vinegar in your milk. All of a sudden you've got curdles of lumps. That's the protein. Instead of being, you know, soluble and dissolved and you don't notice it in the milk, it's a milk protein. If you change the environment for it, you can get it to change. You denature the milk and you get lumpy stuff in your milk. I think that does it for proteins. As always, if you would like to subscribe, don't forget to click on the gecko. One more video in this series, that's nucleic acids, and we'll get to that one soon. Thank you.